So, welcome back to this class. So, previous class I told you that we will be sharing how you can mimic the human brain's electrical activity on a chip and how you can recapitulate some of the events happening inside the brain. So, as a case study, we took an example that something which is influencing substantia nigra, whether it influences the other part of the brains or not. One of the way, without invading or without killing thousands of animals to figure out this, you can do it on a chip, and that is what I was telling you, that where the technology is moving. Now, say for example, use one rodent or you use some kind of a stem cell to develop substantia nigra neurons and hippocampal neuron. Either your source could be some form of a stem cell, or you can use few rodents to collect the cells. Now, what you can do using advanced photolithographic techniques, you can develop, you can lay down the extracellular matrix in the form of a circuit. So, what is your target? Your target is that you have to establish a connectivity between the hippocampal neuron and the substantia nigra neuron. What you can do is, say for example, you have a microelectrode array chip like this. This is tailor made microelectrode array chip. Okay. Now, you create these dotted lines are showing where the neurons can grow, and these are extracellular matrix which are printed upon the electrode surface as well as non-electrode surface. So, these are E's stand for electrode, E1, E2, E3, E4, E5, E6. This is a simple case study I am showing you, and what you see out here are small tunnels. A tunnel big enough to allow a neuronal process to grow, but not big enough for the cell to cross through it. Okay. Now, you take these substantia nigra cells and you plate them here. So, obviously, they do not have much places to go except to sit on top of these electrodes, because the rest of the area is non-cell adhering or non-favorable substrate. So, only those dotted line are the areas where the cells will grow. Similarly, you take the hippocampal neurons and make them sit in this zone, and you allow them to grow. So, automatically once they are growing following this path, they will cross through these tunnels and they will make connectivity. So, as soon as they make connectivity, that means they are forming the synapses. Okay. They have a synaptic connections. Now, once they have the synaptic connections, now they will try to do a electrical activity. So, say for example, this area will start to communicate or vice versa between each other. I mean, this is just an example. This may not be in a real life happening, but just you have to understand how the technology can help you to answer something which was never feasible prior to this. Now, what you do? So, as I told you, these are in two separate chambers with this connectivity. Now, you add the toxin to this. Now, when you are adding the toxin, so automatically the proximity of the toxin is close to the substantia nigra. So, automatically it will affect these cells. Obviously, there is no denial that some of it is going to diffuse to this side, but that will take a longer period of time, right? And that can happen in the brain itself through cerebrospinal fluid, right? Now, you see how this neurotoxin is influencing the electrical activity here. Does electrical activity of this is getting compromised, or how much time does it take? So, as I mentioned, you can do a lot of chronic experiments. Okay. Similarly, you can use this model for another very interesting thing. Say, for example, instead of say different parts of the brain, you want to understand how a neuromuscular junction works. What you can do, you can grow the muscles on this side and you grow the neurons on this side. They will travel and form connectivities, and you can study the neuromuscular junction toxins. So, you can put the toxin on synaptic side, neuromuscular junction side, you can put the toxin on the neuronal side. Similarly, in the case of substantia nigra and hippocampus two-chambered model, you can put the toxin on this side too and see does it influence substantia nigra, and vice versa. So, there are thousands of combinations what you can do, but in order to do so, you have to have an understanding of how to manufacture planar in a large number planar microelectrode array. One, you have to understand photolithographic tools. You have to have a hang of cell culture technologies. You have to have a hang on microsystems fabrication and electronic interface. These are some of the tools, key tools what you need in order to study these kind of systems. So, next level comes how we can translate. Say, for example, in the beginning I told you that there are different ways you can translate these kind of informations. So, as I mentioned, one of the major challenges is that most of this toxin leads to paralysis. So, paralysis means your muscles are not moving, and muscle is a structure which moves. So, if you recollect back, as I mentioned, there are 3-4 events which happens. A neuron secreting, generating an action potential, reaches the neuromuscular junction. There it leads to the secretion of neurotransmitters acetylcholine. Those acetylcholine travels through that small cleft of neuromuscular junction or neuromuscular cleft and jump to the other side and binds to the vacant spots which are meant for acetylcholine binding. Upon binding, they open up the cation channels. Those cation channels led to the entry of sodium influx inside the muscle. Sodium influx entry triggers an action potential within the muscle. So, the first action potential was in the neuron, then there is a chemical transmission followed by a action potential generation by the muscle, and the action potential generation by the muscle leads to activation of the sarcoplasmic reticulum inside the muscle, which was sensed by ryanodine and DHPR, dihydropyridine receptors. That leads to the release of calcium from the sarcoplasmic reticulum transiently before it is being pulled back by the sarcoplasmic reticulum by the different pumps. But that spiking of the calcium plays a critical role in the actin-myosin filament movement, and in that process, the final outcome what we see is a muscle movement, and this whole process is called excitation-contraction coupling theory, where excitation and contraction is being coupled together in this process. Okay. So, now what I wanted to share is this whole process, as I have already mentioned, you can recapitulate in a microelectrode array or using patch clamp electrodes. But now I will tell you a different kind of sensors where cantilevers have been used, because there is a muscle involved in it, and muscle is generating a force, and this force varies. So, if you look at the different kind of muscles in your body, so there are 3 different kind of muscles in your body: skeletal muscle, this is the maximum force generating muscle. Within skeletal muscle, there are 2 different kind of fiber. One is called, which is an extremely less number, less than 1 almost, interfusal fiber, and extrafusal, which is the rest. Then you have cardiac muscle, and most of the force generation is done by cardio, myo means muscle, cyte means cells. Then you have smooth muscles. Now, what distinguishes each one of them is the force. Force generation. So, in other words, the actin, myosin proteins. Now, most of the neurotoxins hit either this or this or this, and smooth muscle, just for you, are the ones which are taking the major one are the digestive tract and other places where involuntary actions are happening. Now, how we can without using animal? So, the question is, without using animals, how we can study the force dynamics, or we can perform the force measurements So, in other words, what we wanted to do, we wanted a animal free system. Now, again back to the technologies I was telling you, microfabrication and electronic interface is the one which comes into your rescue. So, one of the technologies is, say for example, you have a micro cantilever platform. Now, you wonder what is a micro cantilever platform. Okay. Something like you all have seen the bridges, like any kind of bridge, say for example, a bridge like you all have seen Howrah Bridge, which is something like structure which is suspended structure. So, you all have seen in the swimming pool, you have seen the diving board, right. Now, say for example, when a diver goes, so this is the staircase you are climbing and diver is standing here. So, what you experience is that there is a movement happening here before this guy jumps into the water. So, there is a kind of a motion which happens, and you can actually figure out the motion in a very simple way. Say for example, from here, from a fixed spot here, you shine a light here, or you shine a light to the tip of this. So whenever there is a motion, so you see the light will, what will happen, so whenever you are, there is a motion out here, if this is the regular reflection angle and you have a position sensitive detector, whenever there is change, you will see a kind of something like this. So, there will be change theta which is happening. So, technically speaking, you should be able to figure out this change, you can detect this change. Now, if you know this change, there is a way you can back calculate the force. So, that means when you are talking about the force, that means we are talking about how much force this particular individual is generating on top of that diving board, right. Now, this is exactly the principle when I am telling you about micro cantilever arrays. These are suspended. So, imagine each one of them, let me redraw this for you, something like this. Each one of these are diving board. So, there is a cut underneath. Right. Now, if we could grow individual muscles on top of these, and of course, there are two ways we can think of it. Either we, since we are not growing the neurons here, then what we can do is underneath we have electrodes present, which will do the job of the neurons. These are the electrodes which are present there, E, these are the electrodes. Now, instead of the neurons, if we have the electrodes which are present there, what will happen is that you can control their excitation, which leads to the contraction, right. So, in other words, you can actually give an electrical impulse to generate a force from the muscle. Now, think of this situation. Replace this individual with a muscle and replace this staircase out here with an electrode, and underneath you have, and here replace this light with a laser beam, and here you have position detector. In the next class, I will pick it up from here and I will explain how this technology could completely alter the way we are going ahead in understanding one of the most fundamental circuit, which is a potential biosensor tool for the future research. Thank you.